Does stress level influence pyocin production in Pseudomonas aeruginosa? P. aeruginosa is a gram-negative bacterium. It is commonly found in the environment. It infects individuals with cystic fibrosis who are highly susceptible to bacterial infections. As you can see on the graph in the left, P. aeruginosa is the dominant bacteria infecting people with cystic fibrosis. P. aeruginosa undergoes extensive phenotypic evolution in the CF lung. During chronic infections, common modifications include changes in colony morphology, which can be seen on the left. In the center, you can see reduced pyocin production in CF isolates compared to environmental isolates. And on the right, you can see that CF isolates display high levels of antibiotic resistance. In figure A, you can see that under ideal conditions, pyocin production is repressed. As shown in figure B, in response to DNA damage, Rec-A is activated, which cleaves PRTR and allows pyocin genes to be expressed. They are released through cell lysis, and greater than 95% of P. aeruginosa isolates encode one pyocin gene. Pyocin are antimicrobial toxins that inhibit the growth of other bacteria.
Previous studies have used a wide range of mitomycin C concentrations to induce pyocin production. As displayed by the table, the concentrations used have ranged anywhere between 0.01 to 3 micrograms a milliliter. Our first research question was, does the concentration of mitomycin C influence pyocin production in PaO1? And then, what would the optimum concentration be? Our second question was, does the concentration of mitomycin C influence pyocin production in CF isolates? For our materials and methods, we started out incubating our bacterial isolates overnight at 37 degrees Celsius and 5 milliliters of Mueller-Hinton broth. Then we took that bacterial culture and diluted it at a 1:100 ratio and incubated it again for 4 hours. The inducing agent was then added in concentrations of 3, 0.3, or 0.03 micrograms a milliliter, and then the bacterial culture was incubated overnight. The plate displayed in the lower right corner shows one of our pyocin spot assays that we conducted. The clear spots, called zones of inhibition, display where pyocin is being produced.
Our results here help to answer that first research question. Increasing mitomycin C concentration does not increase pyocin production in PaO1, which is the model isolate for P. aeruginosa. Along the x-axis, you can see increasing concentrations of the antibiotic, and along the y-axis, you can see pyocin inhibition. Quantitatively, our graph shows that by inducing greater genotypic stress with increasing concentrations of the antibiotic mitomycin C, the pyocin production by the isolates was not affected. Qualitatively, we are able to observe larger zones of inhibition indicating greater pyocin production at lower concentrations of mitomycin C. This implies that the lower concentrations may be optimal. Increasing ciprofloxacin concentration does not increase pyocin production in PaO1. Quantitatively, the graph shows that there was no statistical significance between increasing concentrations of the antibiotic ciprofloxacin and increasing pyocin production by the isolates as a result. Qualitatively, to the right, we can see that increasing ciprofloxacin results in increased pyocin production, indicating that higher concentrations may be optimal. Currently, our data in progress is showing that increasing mitomycin C concentration decreased pyocin production in the CF isolates. More testing will need to be done to continue to measure this response. In summary, increasing levels of mitomycin C and ciprofloxacin do not significantly influence pyocin production. Lower concentrations of mitomycin C may lead to greater pyocin production due to greater cell survival. And finally, lower concentrations of mitomycin C may lead to increased pyocin production in the CF isolates.